very much, Penny. So very excited to be here, realising that the synergy with Muscular Dystrophy Queensland's just been growing stronger and stronger. If we show you the next slide, my area of interest covers all of the neuromuscular diseases. I guess I've focused on motor neuron disease, but that actually is the hardest disease because we really don't understand the cause. Whereas for muscular dystrophies and other muscle diseases that are supported by Muscular Dystrophy Queensland, we're getting a much better idea as to the actual cause. And that means we're a lot closer to potential treatments. So neuromuscular diseases covers anything that's basically outside the spinal cord that affects movement and sensation, coordination and fatigue, and how we use our limbs is covered by neuromuscular diseases. So on the next slide, the area of interventional trials, I've just covered there the broad groupings of the diseases that I'm involved with. And so it came about that we were originally focused at Royal Brisbane and Women's Hospital, but it became clear that you need to be able to work across different centres which have advantages and disadvantages. And a nice example would be that Royal Brisbane is just too busy as a public hospital. Most of its traffic is people who are admitted through the emergency department. So it's really hard for early phase studies where you're wanting to collect samples or do measurements after hours. You really had to be a bit more flexible. So there's a structure called Nucleus Network that allows that, but Wesley Hospital has also been very supportive of neuromuscular trials. And the University of Queensland and QUT have also been very involved with clinical trials. So on this slide, the first part of the talk, I'm going to focus on the muscular dystrophy clinical trials that we're involved with. And then if we've got time, I might mention the Pompe disease and inclusion body myopathy clinical trials that we're involved with. So next slide. So I guess this is a broad brush on what happens with a gene discovery. And in the past, a lot of drug trials have focused on drugs that are already being used for other purposes, and that's called repurposed drug trials. And that's because you don't have to spend 10 years proving that the drug's safe. In humans, you've got a pretty good idea what the dose is. But most of those trials have really made not much difference. I guess myotonic dystrophy type 1 has been an area where many of these drug treatments have been tried. So the bigger focus over the last 5 years has been disease-modifying therapies by actually targeting the gene. And I will try not to confuse you about what gene replacement and gene editing means. And those are actually the true gene therapy trials when they're actually targeting DNA. Whereas DNA, which is in the nucleus, becomes RNA, which eventually gets out into the cytoplasm. And other drug therapies, which fall under genetic therapies, are these RNA or ASO types of therapies. And finally, if you do know what the actual gene and what the RNA is doing, you can target the protein directly. And so some of the clinical trials are more focused on the protein. So if we go to the next slide. So these therapies are actually happening and are actually getting to the stage of approval. So a very rare form of motor neuron disease is when you've actually got a known gene and you can target that gene and that drug appears to work. There'll be people online who are aware of spinal muscular atrophy, and the drug therapy for that needs to be given intrathecally into the spinal fluid. But there is a reasonably large number of Queenslanders with spinal muscular atrophy, both adult and children, who are receiving nusinersen regularly for the treatment of their spinal muscular atrophy. And also in areas of neuropathy, which probably be a little less covered by Muscular Dystrophy Queensland, but some of those neuropathies, in particular one called hereditary amyloidosis, again, there is now a gene therapy. So next slide. So just to put a little bit more detail in the actual diseases. So the actual gene therapies where you're using something to get the gene into the nucleus. So that's been possible, and it's usually a one-off therapy, but Duchenne's muscular dystrophy, myotonic dystrophy type 1, and Pompe disease. And if I put them up in red, it's the diseases where we've actually got clinical trials happening. And then I mentioned before the ones where you're actually changing gene expression, so you're acting more on what's called RNA, myotonic dystrophy type 1 and FSH muscular dystrophy. And then some of the other muscular dystrophies have gene editing. So next slide. So this is all rapidly expanded in the last 5 years. So if you are reading reviews before 2020, most of this is just going to be missing. It's been interesting to see that the technology that gave us some of the COVID vaccines based on mRNA are actually leading to some of these therapies. So next slide. The muscular dystrophies aren't simple as just saying, well, it's a gene that does X, and therefore I can treat it with Y. So people with these diseases who are online will know that, myotonic dystrophy and FSH muscular dystrophy are more complex than just saying there is one gene that's causing a problem. What both of them share in common is they lead to the dysregulation of a number of other muscle genes. So they actually don't fit in the textbook that doctors learn about what the actual muscle complex for how the muscle fibers move next to each other. These genes are actually these diseases, myotonic dystrophy type 1 and FSH, are a little more complex than that. But both of them, you very clearly know what the gene is. So for most people with FSH, it's called the DUX4 gene. That's the problem. And with myotonic dystrophy, it's the DMPK gene. Now, not everyone with myotonic dystrophy is type 1, and not everyone with FSH is type 1. But the clinical trials have largely been focusing on the type 1s in both of those. So next slide. So I've put up here the names of the companies, and if you put in the words myotonic dystrophy and Vertex, for example, and clinical trial, you can actually bring up the clinical trial information, because these are all large international companies that will register on a site called clinicaltrials.gov, and they'll provide information and updates on their clinical trial. The only thing that's a little bit fluid is that they're essentially biotech companies, and they may be taken over or, so to speak, swallowed up by larger companies. So, for example, in the last few weeks, Arrowhead is actually changed, and it's now part of a company called Sarepta. And people who know the Duchenne muscular dystrophy know that Sarepta has been one of the drugs that has been developing one of the drugs for Duchenne muscular dystrophy. What this slide also can show you is that there is a large number of companies sort of competing, but they're sort of not. They've all got quite different mechanisms of action, and they're targeting things in different ways. So in my earlier slide, I sort of had there that you had gene replacement, gene editing, or targeting proteins. So they're sort of different mechanisms. So it's easy enough for us to be involved with multiple companies and not really have any conflict of interest. So as I said, we're involved in 3 active clinical trials in myotonic dystrophy type 1, and probably starting up a 4th one very soon. Next slide. Similarly with FSH muscular dystrophy, there are 2 active clinical trials. Vertex has been going for a little while. Souffle is a US company that is just about to start. And Javina is also a US-based company that's going to be recruiting within the next few months as well. Next slide. So let's start with this first trial, which as of last week is now no longer called Arrow DM1, because as I said to you, Arrowhead has been taken over by Sarepta. But what I'm wanting to show you on these slides is just a little snapshot of the basic ideas of the clinical trial. So, for this one, it's working on that RNA. So as I said to you, the true gene therapies are acting on DNA. The ones that are acting on RNA, usually you have to keep giving the drug at different periods, monthly, 2-monthly. And, the word SAD means a single ascending dose. So that's usually when you're trying to find the safety of the drug. The MAD means multiple ascending doses. So that's when you're usually trying to find exactly what dose of the drug is working. The trials usually start off as what's called phase 1-2. And then usually there'll be an extension phase where they'll continue giving the drug afterwards. This one here is an IV infusion. I think it's about 30 minutes that the infusion takes. Many of the trials are a little biased against people older, and 65 years for this trial. A couple of the others are even lower. In Australia, it's very easy. People have a diagnosis based on a genetic test result these days. The outcome is to use muscle biopsies as well as functional measures of what people can do. I'll show you a picture of what the muscle biopsy is. A 10-minute walk test is a fairly standard outcome, how far you can walk in 10 minutes. And the video hand opening is a pretty novel way of assessing myotonic dystrophy. And in that, our research nurses watch patients opening their hand as quickly as they can. You've got to squeeze it tight for a few seconds and then open it as fast as you can. And that gets videoed, and then all of it gets uploaded to a central repository over in the US, or with the main place where these trials are being conducted, and someone is able to do it in an unbiased way and say, well, it took them 3 seconds or 5 seconds. So those are the outcomes. Next slide. Oh, there's the picture of the muscle biopsy needle. These clinical trials have sort of changed how we consider taking muscle biopsies. It used to be, and there'll be people online who are aware of a muscle biopsy being a scar that could be 3 to 5 or more centimeters. But this needle involves, it's largely been painless. I think, again, there'll probably be people online who are part of these clinical trials, but the needle is placed in the muscle after some local anesthetic. For most people, I wouldn't say they're saying the muscle biopsies as a major issue. Next slide. So this one is also showing that same company doing the FSH clinical trial. So there's roughly about 9 or 10 people with myotonic dystrophy in the first Arrowhead study, and about 9 or 10 with the FSH who are involved in the second study. First study is happening at Royal Brisbane. The second study with FSH is happening at Wesley. This one is also fairly similar to the myotonic dystrophy study. Again, it's using muscle biopsies and strength assessments. The big difference is to get into this trial, you've got to meet an inclusion criteria where you get this whole body MRI and they assess what's called the fat-free muscle. So that has been an issue for some people to get into the study. In general, earlier phase studies looking a bit more at the safety and the dose are quite happy to recruit people across a range of the disease, whereas later phase studies are probably wanting to more carefully evaluate people based on function. So they're looking at people who are probably at earlier phases of the disease. So next slide. And this is the Vertex myotonic dystrophy study that is also happening at Wesley. Again, it's had different phases. It's now at a phase where they're looking at their 6th dose of what to assess. But people who are getting lower doses in earlier phases should be eligible for later phases where you're getting the dose that they're studying. So next slide. So the gene therapy study that we've been doing at the Royal and to date have recruited 1 person, but this has been way more complex because this is actually using a virus to get into patient cells to try to modify the muscle cells using this virus as a vector. So 1 person's had that at Royal Brisbane. So far, that's not, it's relatively early with that clinical trial, but we're trying to assess that over a full year. I think there's a number of things that have been helpful for us to try to, with all of those clinical trials, is a structure that we've developed, and we talked with Muscular Dystrophy Queensland just recently about how we could collaborate a bit more with our structure called the Nerve Connection Foundation. But one of the things, by having a well-structured number of doctors and nurses, we've been able to recruit patients all across Queensland in these clinical trials. So we're really not focusing just on the southeast corner like we were with motor neuron disease. We're really, in these neuromuscular trials, we're really hopeful we can recruit people right across the whole of Queensland. Next slide. I'll skip through these 3, so just jump through these, but this is basically assessing, and next slide. So that was, next slide after that. Yeah, so that basically has been saying fairly similar outcome measures. Do these drugs make a difference? So I'll jump through to another, next slide. I'll talk another muscular dystrophy. It's a good example of where we lie. A patient with this relatively rare muscular dystrophy, limb-girdle muscular dystrophy. They approached us because they'd heard of a clinical trial in the US. And because we had a structure to support clinical trials, we said, yes, we'll be happy to try to help. And before we knew it, we've got 7 people and possibly the largest site for that. It has been a successful clinical trial, so it does mean that we're likely to see an audit by the FDA to say, well, has the trial been conducted correctly, which is part of a normal process in trying to get a drug approved. So this is actually more targeting a protein. When I talked before in that slide about how drugs work, for this one it's an oral therapy, and people are assessed again with muscle biopsies and again with functional measures. This type of muscular dystrophy affects the heart muscle, which is obviously, one of the most important muscles. So if it's going to work, that would be actually quite exciting. Next slide. So up the top there, you can see our fellow, Namrata, performing a muscle biopsy. There's nothing special about it. People come in and and walk out, but it's very much a key outcome measure, because you can look in the muscle about how the drug has actually affected the muscle, and they use the word splicing indexes and how the muscle has changed. Next slide. So that interim analysis of that trial's actually only just been released a couple of months ago. Next slide. And relatively small, because it's a relatively rare muscular dystrophy. But first of all, you have to work out what's called a power analysis to say, well, have I recruited enough people to be able to prove my drug works? Most clinical trials work on the idea that 2 to 1 or 3 to 1, so that 2 or 3 out of 4 people will get an active drug, but some people will get a placebo. And then you'll have a period where the drug is being assessed by the placebo, and then everyone will go on what's called an open label extension. And what's quite exciting about this trial is that it appears if the drug works, well, you can say, well, let's stop the study and put everyone on the active drug. Because if the drug truly works, well, people shouldn't be on a placebo. And so what's happened is that the first 12 months has been analysed, and the idea is that people don't stay on the placebo for the following 24 months. Next slide. So again, when you're doing a clinical trial, you've sort of got to prove the drug works, and there's a very rigorous way to do that. You can't claim your drug works but not have the evidence to back it up, and you've got to have your populations matching. And so this is just sort of showing you that most people were actually adults, relatively equal gender, but you've got to be able to cover all races, because you can't just say my drug works in white people. And then you can say, well, what was the gene and how did it affect the different gene? So next slide. And this was the outcome measure that showed a very clear benefit for people on the drug by looking at the muscle biopsies and showing that it led to an improvement in the alpha dystroglycan, which is the protein that's affected. Next slide. And you can basically look, with any clinical trial, you have what's called a primary outcome and then a secondary outcome. So basically, you've got to prove your primary works, and then you look at your secondary, and the secondary should back up the primary. And so that's what's been able to be shown in this clinical trial, that it all does back up. So next slide. And people in this audience will know that the CK is a very important measure, and so the CK level clearly improved impressively in the people. So next slide. And again, you're looking at people's walking ability. Next slide. And time to complete 100-meter. There was a clear benefit for people. So next slide. And also that's looking at lung function, showing that that's also improved. So next slide. And with any clinical trial, the important thing is also, are you seeing side effects? And so there's, no clinical trial is totally free of side effects. People in placebo arm will also report side effects. And so any muscular dystrophy trial, people are going to have falls. And if you fall, you're going to be recorded as an adverse event. So, headaches, nausea, arthralgia. So next slide. So, that's quite exciting that drug trial appears to show a benefit at the first stage of analysis. Next slide. So I'm just going to cover a little bit on inclusion body myopathy, because I know there's a number of people who are supported by Muscular Dystrophy Queensland with this condition. It's quite a common condition, increasing with age, a bit more common in men. And there's been 2 clinical trials that we've been involved with, one with a drug called rapamycin and one with this drug called ABC008. So next slide. So the interim analysis, again, just from the last few weeks, is not showing a definite improvement. Next slide. But people who are a bit earlier in the disease, so there's a score where you can measure what your level of function is, called the inclusion IBM-FRS, inclusion body myopathy functional rating scale. And people who are with a higher baseline score certainly might have some benefit, and that's part of an ongoing analysis. Next slide. So people who started with a higher score possibly showed a benefit from this drug called ulviprubart. And what that also shows in any clinical trial is you look at a couple of different doses to see if a higher dose is better. And in this case, a higher dose was no better than the lower dose. Next slide. Yeah, so we've also been looking at trying to see better markers of disease. And so one of the key ones is we're using MRI and PET studies at Royal Brisbane to look at can we come up with better markers of whether the disease is doing anything. So in that study, inclusion body myopathy, there's a number of people who are having regular PET studies to assess that. Next slide. People in the audience will be aware of the drug rapamycin, sirolimus. Very much, this had a large number of people across Australia involved in it. They haven't reached the stage of interim analysis. So they recruited a reasonably large number in Australia, but they wanted to prove the drug worked across multiple sites, and they haven't finished the recruitment internationally. So that trial, we don't have any results to share for you. So next slide. People can continue to take that drug if they wish. So Pompe disease is another muscle disease, relatively rare. I've got a number of patients. They're probably not as much involved with Muscular Dystrophy Queensland, but could be. And they're another nice example of a clinical trial that clearly showed a benefit. So the drug Nexviazyme was a clinical trial that we ran at Wesley that clearly showed a benefit. There was another clinical trial with a company called Amicus at Royal Brisbane that has also shown a benefit for this muscle disease, but the drug is not yet approved on the PBS. But there are a number of people in Queensland who are receiving that on compassionate access. So by understanding the mechanism, you can sort of target the end result. But this is a disease where we've been largely targeting the end result, the protein, similar to what I talked about with that limb-girdle muscular dystrophy. Whereas the earlier studies in myotonic dystrophy, FSH muscular dystrophy, we're targeting the gene. Next slide. I really hope I'm not confusing anyone with the different terminology, because it is a bit challenging. So the Pompe disease, the hope is that by understanding the gene, you can actually have a proper gene therapy targeting the gene. So we've also been involved in those clinical trials to understand, are people developing antibodies against the vector that we're going to use to deliver the gene into the cell. Next slide. So finally, there'll be people online who have even rarer muscular dystrophies where the actual number is small, but you'll be aware that if you contact people overseas, that they'll have registries or they'll be doing observational studies. I'm aware of this dysferlinopathy, and the University of Newcastle in the UK are doing a registry. It might well be that, you know, a clinical trial, if you know what the gene problem is, you might be able to have a clinical trial without a large number of patients, that you can have targeted gene therapies. That is very much on the horizon. So there is quite an exciting time ahead for people with muscular dystrophies, I think, particularly where the gene is clearly recognized. Next slide. Yeah, so basically, to run clinical trials in these diseases, you've got to be able to target and have mechanisms that can give the drug in many different ways. You can have different structures that can keep patients overnight, that can perform the muscle biopsies or lumbar punctures. And next slide. And also, you've got to have a whole team involved. You've got to have what's called research fellows. Often they're doing a research PhD. See if we just move to the next slide. Sorry, I've left off the slide, which is important because it shows the research nurses and research fellows. And, you know, by building those teams, we've sort of built them both at Royal Brisbane and at Wesley. And in some ways, they're quite separate. But I guess by having an overarching structure called the Nerve Connection Foundation, we're funding them across both centres. By linking in with the big universities also has been very important, but also having a place where you can do earlier phase clinical trials like the Nucleus Network is very much a way you can target many different facets of these diseases. Because I think they're all relatively rare, but combined they're not rare. And combined, you know, we would probably have over 50 people living with these diseases involved in clinical trials. So it has been a real growth and a really exciting part of neuromuscular diseases, because most people here online will be aware that really there hadn't been any of these therapies for a very long period of time. So I might stop there and allow a little bit of time for questions, I think. But I hope I haven't confused you, and I hope I've given you a snapshot of the exciting times ahead. Thank you.
Thank you, Dr. Henderson. That was very interesting, and as you say, exciting to hear that there are so many developments and so many different neuromuscular conditions. So we'll move into a time of questions now. I've got a few that were sent to us initially, and then if you'd like to have a question answered, please feel free to pop it in the chat. So the first question that came through was, should a person with myotonic dystrophy have an ultrasound of the veins in their arms and legs if there is a loss of function?
So imaging isn't the best outcome. So if the reason you have an ultrasound can be to see if you haven't got a clot in your leg and making sure you don't have a deep vein thrombosis to affect the level of function, because that needs to be treated separately. Ultrasound's not really being used in the outcome measures. It'd be obviously a nice non-invasive way to assess things. It can show differences between nerve and muscle disease and comparing that with normal, but it hasn't really been used in clinical trials.
Thank you. The next question, is it considered safe to regularly take supplements such as canine pepper and turmeric?
Yeah, so you've got to look at the individual supplements to know that. One example would be if you take too much pyridoxine, that can actually cause sensory damage to your nerves. So some of them you can't take too much. In general, if you do your research, you can see whether they're causing more issues. Most of the time, for those ones you just mentioned, they shouldn't be. They should be safe at any dose. A nice example too is magnesium, which helps many people and usually can be regarded as able to be taken in whatever dose you wish to take. But clearly, if you take too much magnesium, your bowels become a bit looser, so you're not going to be taking too much magnesium. But in general, most supplements are reasonably safe.
Thank you. And the last question that I have that was pre-sent, in which you've already talked to, but could you go over again, how extensively is research currently focusing on myotonic dystrophy?
So myotonic dystrophy has been the one that has led the charge. I think if it's pediatrics Duchenne muscular dystrophy, the myotonic dystrophy, it's a big undertaking to be involved in a clinical trial, particularly, it's easier when people have paved the way beforehand. I always think, a lot of people, the bravery, particularly motor neuron disease, where, very much people are saying, well, it may not help me, but it might help other people. I think it's a bit different for these because there is hope about these clinical trials, but myotonic dystrophy is very much the one that is leading the charge, I think. Does that answer the question, Penna?
Yeah, I think it does. Thank you. Is there any other questions, Sarah, that have come through on the chat that for Dr. Henderson?
Yes, we have one from
Emma. Emma's just Joshua's just put, if someone participates in two trials, A and B, but trial A has an open label, would the participant still be eligible for the open label? And then Emma's just commented an important component of that. So you would sign up for one clinical trial at a time. And the most important thing about a clinical trial is the patient information and consent form. You don't do anything until you sign a consent form. But if you're involved in a clinical trial, you can withdraw at any point in time. You can go on another clinical trial at any point in time. The second clinical trial would usually say, well, we've got to have a washout period from the first clinical trial, which for most drugs is roughly a month. That you couldn't be between one. You'd have to wait a month between clinical trials. It's that difficult balance that you might be taking a placebo. So that's part of the bravery of putting your hand up for clinical trials. And I think it's really important that you need to know whether there is an open label extension at the end. But Emma's in the chat box is also for people who are potentially thinking of clinical trials, Emma's put that quite nicely in the chat about, you need to be fully informed when you're going into putting your hand up for a clinical trial.
Thanks, Rob. There's a few more that have come through. Someone said, sorry, I missed the start. Is there any trials out for Ulrich CMD?
Yeah, so CMD is usually meaning congenital muscular dystrophies. And so Anita Cairns is giving a talk after me, and she's very much involved in clinical trials through the Queensland Children's Hospital. I guess there's a big divide between children and adults, and I guess we're hopeful that we're going to be able to run clinical trials in collaboration with Anita, particularly in that Duchenne's muscular dystrophy area. So I'm not so aware of the Ulrich one.
Thank you.
Someone else also says, how much longer will it be before the general public have access to the treatments in trial? Yeah, so if you use the example of Pompe disease, if the trial clearly works, then the process can be relatively quick. If the trial doesn't clearly work, it clearly, the people who have to approve these drugs, and their expenses are huge, they're going to be a bit less likely. But if you use the example of that Pompe disease with the drug Nexviazyme, we finished that trial at Wesley, and within 2 or 3 years it'd been approved. The two processes that usually fund it are called the high cost drug committees of hospitals, and the federal government has this process called the life-saving drug program. So, for Pompe disease, that approval went through the life-saving drug program and was approved quite quickly. So it'll be interesting with the process, with the approval for this other limb-girdle muscular dystrophy 2I, because it's, if you're trying to really get your drug approved, you usually have been historically going to the FDA in America and getting approval, and a similar process is the EMA in Europe. But for really rare diseases, the FDA has just gone a bit funny in the last couple of years. So we're just not sure about that process. So that'd be the only issues I'd possibly comment on. But the idea, if the trial works, we would be really hopeful that people push, and lobby groups from patients also matter. The TGA in Australia would listen to people living with the diseases. Can I just say one final thing is that, if people do have these muscular dystrophies, then what we would really like to be linking with Muscular Dystrophy Queensland is a proper registry of people with those, so that we can link with Muscular Dystrophy Queensland and say, look, we have a clinical trial that might be one that of interest to you, and if you're part of a registry, then that would be the first way, because if we don't know about you, we won't know about you, and you won't know about the clinical trial. So I think it's really important now to try to be part of registries or databases to know about whether clinical trials are there for you. I think Emma is also, through FSH, she's been a major leader in this area.
Yeah. Thank you. And that's certainly something we'd be keen to talk about. Thank you, Dr. Henderson.
Thank you.
Thank you.